I'm Amanda Celli, as I hope all of you know by now, because I get up here every week. So I'm going to be presenting on anxiety. So the name of my paper is A Cortical Circuit for Sexually Dimorphic Oxytocin-Dependent Anxiety Behaviors, or a mechanism behind gender-specific behaviors in mice. So just a little bit of just one statistic about anxiety is that 18% of the population suffers from anxiety disorders. So just some terms that you need to know. The first is oxytocin. Oxytocin is a hormone that contributes to social behavior. And oxytocin binds to oxytocin receptors, just a little bit of bio one. It binds to oxytocin receptors to create that social behavior. And oxytocin receptors are found on oxytocin receptor interneurons. Oxytocin usually contributes to maternal care, social cognition, things like that. So the next thing you need to know is optogenetics. Does anybody know what optogenetics is? Okay. Optogenetics is altering genes, and what it does is it makes neurons light sensitive. So if you turn on a light, it then makes the neurons activate, and if you turn off that light, the neurons are no longer activated. So light on, the neuron is on. Light off, the neuron is off. So here's a little video to kind of explain. So can you turn off the light? Really quick, just for this. Yeah, so I don't know, hopefully most of you can see, but this is the activation light that I'm talking about. So here is the video. So this has normal behavior. The light is not on the mouse, and then it just turned on, and now it's completely changing the mouse's behavior. It's going in circles. It's like activating the motor cortex or something. And now that light is activating neurons to make it run in circles. Okay, so now back into my paper. Thank you. I didn't even need to ask. So the first thing that they wanted to look at is how activating these oxytocin interreceptor neurons would affect the behavior of females and males. And what they found is that when these oxytocin receptor interneurons were activated, female mice had altered sociosexual behavior. And the way that they measured sociosexual behavior is how much time they interacted with another mouse compared to a Lego. So the opposite of hanging out with other people is hanging out with Legos. So, females, you can see, had an increase in sociosexual behavior when the light was turned on and when the neurons were activated. But when males, when their oxytocin receptor interneurons were turned on with optogenetics, there was no change in their sociosexual behavior. But then in males, what they found is that when they turned on those oxytocin interreceptor neurons, they were less anxious, and when the oxytocin receptor interneurons were off, they showed extreme anxiety. So an EPM is a kind of like a box on two sides and then open arms on the other sides, so that the time that they spend in the little boxes, they're more anxious. The time that they spend on the open arms, they're less anxious. So you can see that the time that they spent on the open arms is highly increased, which means they're less anxious, the males. But females, their anxiety behavior was not altered. So what we found out also is that when oxytocin receptors on the oxytocin receptor interneurons are not present, so that's what the right-hand side means. When they're not present, it's almost as if the light is off again, and they are more anxious, and they didn't spend as much time in the open arms. But females were not affected. So this kind of raises the question of, okay, so this is the same receptor, the same neurons, but why are they acting differently? So the easiest way to start to see it is to build this pathway. So you can see here, the oxytocin leads to the oxytocin receptor and on the oxytocin receptor interneurons, which affects social behavior. But for males, oxytocin and oxytocin receptor interneurons affect anxiety behavior. Can someone repeat that back to me? Jenny?
So basically for the female mice, the oxytocin receptor might activate and leads to sociosexual behavior, but for the male, it leads to anxiety.
Yeah. So either turning on or off oxytocin receptor interneurons changes behaviors specifically in males or females differently. So next I wanted to see, okay, so these are the same neurons, but why are they acting differently? So they looked into the different layers of the brain, and so here you can see it's almost like the Earth and how the Earth is like concentric circles and like it leads to the Earth's core. It's similar, and 1 is the outer, and 2, 3, which we are going to be focusing on, and 5, which we are also going to be focusing on. They're just, this is just to display like the different layers so you can see it. So what we found is that the layer 2, 3, and layer 5 were activated the same amount. So female and males had the exact same response to the optogenetic activation in those layers. It's not like they were missing neurons or those neurons were not firing. They still have those 2 layers with the same amount of neurons. They wanted to see, okay, so maybe 1 is more activated in males and 1 is less activated in females. So what they found is that layer 5 is much more activated in females, and layer 2, 3 is much more activated in males. Are there any questions? Yeah?
How are they measuring activity?
They're measuring activity by the amount of current. They're measuring the current.
Can you just explain these?
It's inhibitory postsynaptic current. So the higher the amount of the higher bar means that's what layer is being more activated when oxytocin receptor interneurons are activated. So when oxytocin receptor interneurons are activated, layer 5 is more active and has a higher current than males. And males have a higher current in the 2-3 layer. Yeah? Okay. So what we figure out though is that, okay, so oxytocin receptor interneurons affect layer 5, which then affects sociosexual behavior. And oxytocin receptor interneurons in layer 2, 3 for males affect anxiety behavior. Bless you. So here's just one more term that you need to know. It's CRH and CRHBP. So CRH is a neurotransmitter. It's the hormone for anxiety and stress. And so it's released during times of stress to coordinate the behavioral response. And CRHBP is a binding protein. And so what CRHBP does is it inhibits CRH from binding to the receptors. So it binds to CRH and inhibits its stressful effect. So can someone explain to me what CRH is?
CRH is a stress hormone, and then CRHBP inhibits that so you don't get those effects of stress and anxiety.
Yes. So what they wanted to see, okay, so they knew that these 2 layers were being activated, but what's different between those layers that's causing these different behaviors? So what they did was a protein analysis, and so they figured out, okay, so what's in each layer that's making it different? So they found that the CRH receptor that the CRH binds to is only in the 2-3 layer, but it's not in the 5 layer. So what implications would the CRH receptor location have on these gender relations? Yeah?
You said that males have a higher amount with the
So that's very close. So CRH, this is really complicated. So the location of CRH is saying that the receptor is, what we just need to know is that the receptor is in layer 2-3. So it's possible that something is being affected, and that's why anxiety behavior in the male mice and whose brains are being activated in the 2-3 layer have this anxiety effect, because the CRH receptor is in the 2-3 layer. So the female pathway stays the same. We still know that the oxytocin receptor interneurons change sociosexual behavior. But in males, we now know, as I said earlier, is that the CRH receptor is in the 2-3 layer, which then causes anxiety behavior. But how does the activation of oxytocin receptor interneurons affect this anxiety behavior? So they still have more work to do. So what they wanted to see, okay, so what happens when we increase CRH? What happens when we increase CRH and block these receptors? What they found is that females did not respond differently between the control, the added CRH, the added stress, or the CRH and the CRH receptor blocker. Meanwhile, the males, they had an increase when CRH was added. But what would happen when you block the receptor of something that you're trying to bind to? What does that mean? What can't bind?
The CRH.
Yeah, so when the CRH can't bind, that's why it's back to the control level, because the hormone is not binding to the receptor to create that stress. So this is a generalized question, we don't know yet, but what could be possibly causing these gender-specific differences in response to the CRH? Any ideas? Yeah?
Like between females and males?
Yeah. Yeah. So, are there any other ideas? This is an open question. There's no right or wrong answer. That's okay. You can just think about it. So, what we wanted to see is, okay, what happens when we inhibit that binding protein that I talked about earlier? So, CRHBP. Can anyone remind us what CRHBP is? Yes.
It inhibits the CRH, which induces stress.
Yes, so CRH induces stress, and so when CRHBP binds to CRH, it can't induce that stress. So what we found out is that this is the control, these black lines are the control, and this is once again an anxiety study, so the more amount of time that's spent in the open arms, the less stress that they are. So the lower the bar, the more the stress. And so what we found out is that when CRHBP is knocked out and it cannot bind to the CRH, there is more anxiety in males. They have more anxiety behavior and do not go in these open arms. Meanwhile, females, their anxiety behavior is not changed. So what we found out though is that the CRHBP is now also affecting anxiety behavior. So what we found out is that oxytocin receptor interneurons are creating the CRHBP, are signaling for the CRHBP to be made and to inhibit anxiety behavior. So oxytocin receptor interneurons are that key step to lower anxiety behavior. Yeah?
What's the difference between that selection of graphs and the one before with the open arms?
Say again?
What's the difference between these graphs and the ones that we had before with around the time the open arm anxiety change?
So, these? No, sorry.
Yeah, these ones.
These ones?
Yeah.
So this one is with just the, this is the activity of the oxytocin receptor interneurons. This is with optogenetics, and optogenetics either turning on those neurons or turning off those neurons. This one is with either an elimination or a total normal receptor on those oxytocin receptor interneurons. And then, sorry, and these are either the binding protein being there or the binding protein not being there. So it's 3 different pieces to the same puzzle. So, like I said earlier, now we find out that CRHBP is inhibiting this anxiety behavior, and the oxytocin receptor interneurons are key so that CRHBP is being signaled to inhibit anxiety behavior. But now, we still don't know why females' anxiety behavior is not being changed. So what they want to look at is, okay, so maybe it's the amount of CRH. And what they find is that females have a significantly higher amount of that stress hormone. So these are males, this is female. They have about a twofold the amount than males. And this is like the average amount in females. So what we figured out is that while males have this integrated pathway, females have 2 separate pathways for this anxiety, because what they found out is that oxytocin receptor interneurons, when they signal that CRHBP to be made, there's just too much of this stress hormone for the CRHBP to bind to all of that stress and to eliminate it from being from attaching to those receptors and binding and causing that stress. So does anyone want to try and explain this to me? This graph. Amanda?
Okay, so the females have 2 different pathways. One where the oxy interneurons affects their level 5, which will affect their sociosexual behavior. But they also have much more CRH than males, so when the oxy interneurons release the thing that's supposed to stop the stress, they have way too much. So in females, they're unable to lessen the stress. Therefore, they have anxiety.
Yeah. So that's why it's not changed when the oxytocin receptor interneurons were being activated, or when the receptors were changing on these oxytocin receptor interneurons, because there's just too much CRH for that binding protein to affect that stress. So this is the diagram that the paper made, which I thought was too complicated, so that's why I made my own, but just here's a visualization of it, so if you guys wanted to see it. So just a summary. Oxytocin receptor interneurons are essential for male anxiety behavior and to moderate that anxiety behavior. While females, they have just so much CRH that they don't have that same pathway. They don't have that same anxiety pathway that works in males. So, conclusion. So, by identifying gender-specific or sex-specific circuits in the brain, we can identify different gender-specific treatments, and that's really it. So a big thanks to all of these people. Some of them have left, aka Mr. D, Sydney Stein, Mark Strockman, but you can see all of their beautiful faces up here on the board. So if you have any questions. So are there any questions? And you can challenge me. This is my 3rd time presenting. I don't know if I said that earlier, so you can ask me any and all questions, and I'll try and answer. Oh, yeah?
So, you said that it was in the 5th layer of the female?
Yeah.
So is there 5 layers also in the male? Or is it like why is it in the 5th level in the female and the 2nd layer in the male?
So, female and males have the same brain composition, their layers are the same, but it's just that these they're activating different things, because this oxytocin receptor interneuron, when it signals that CRHBP in males, it then affects the 2-3 layer, because that's what modulates anxiety because of the CRH. Meanwhile, in females, they don't have enough of that, so it just, that makes sense. Yeah?
When you began your presentation, you said that 18% of the population experiences anxiety. What population were you talking about?
I think it's I just got like Wikipedia, but no, it's 18% of the U.S. population experiences anxiety disorders.
Human population.
Yes.
And is there variance between men and women with that, male, female?
Based on this study, it's possible that there is, but I didn't look specifically into male and female because I just didn't. Sorry. Any other questions? Okay, so next week we have one of our very own, again, Matt Stanton, presenting on the biology behind caloric restriction. So if you guys want to come out next week and support him, that'd be great.